It's my pleasure to introduce Weimin Bi. She works at Baylor College of Medicine, and we haven't met before, but I've followed her work for a while, so I'm excited to introduce her. I have fondness in my heart for Baylor because we did sequencing there a long time ago, back when I was very excited when they went from 35 base pair reads on the solid up to 50 base pair sequencing reads on the solid sequencer. That was a big day. But so Weimin Bi, she's an associate professor in the Department of Molecular and Human Genetics at Baylor College of Medicine. And through a professional service agreement, she's also the division director of cytogenetics at Baylor Genetics. She did initial training in China and then did her PhD at University of Texas at Houston. And she has a long history of structural variant analysis, including some really great work on Smith-Magenis syndrome and chromosome 17 several years ago. And her research interests now are neurodevelopmental disorders, identifying and characterizing rare genetic variants that contribute to those, and then also improving clinical diagnostics using cytogenetics and looking at structural variants. And this paper she's going to be talking about today is really a great summary of the complexities of CNVs and autosomal recessive human disease in a large group of subjects using multiple different technologies. And so I think a lot of people should enjoy it. So Weimin.
Okay, let me get the share screen. First, thank you, Sherry, for the invitation. Also, thank you, Elliot, for the nice introduction. It's really a pleasure to share our experience on the detection of autosomal recessive copy number variants here with you. First, I need to have this disclaimer. I'm like Elliot explained, I'm a faculty in Baylor College of Medicine. I serve as a lab director in Baylor Genetics Laboratory. That's a company. So the result on our research on autosomal recessive CNV has been recently published in the Journal of Genetics in Medicine early this year. And Bo here is the major player of this study. He did data analysis and also wrote the manuscript. The data we analyzed are from the accumulation of many years clinical work by CMA team and the West team. I also need to point out that the leaders for launching the CMA and the West in clinical diagnosis, Dr. Bade, Dr. Chang, and Dr. Yang. Let me start with the introduction of copy number variants. I know you all know it well, but I wanted to emphasize that the size of copy number variant is very variable, from 1 KB to a few or more megabase. And because it's a genomic segment, it can involve one single gene or hundreds of genes. As you can see below, the copy number gain identified in the FOXG1 region. Sorry, let me see, okay. Let me hide this part. Sorry, I wanted to get rid of something, but okay. Okay, now I can see better. The bottom part is the example to show you the copy number gain detected in the FOXG1 region. The small one is only 88 KB, but the big one is more than 5 megabase. And the smaller one only involve one single gene. The big one may contain multiple genes. CNV can be benign without any clinical consequence. However, some CNV can cause rare genetic disease. Those genetic diseases are called genomic disorders. Usually, the CNV that cause genomic disorder are large, bigger than 100 kb, and chromosomal microarray analysis is a very good way to detect big copy number change. It can provide genome-wide detection of both gain and loss. As you can see in the right side, copy number gain in 16p13 was detected, and also in the same time, copy number loss in 16p11.2 was detected in the same individual. CMA was first implemented in clinical genetic diagnosis around 2004. This paper is a very important paper. Is published in 2010 by Dr. Miner and others. This paper performed a metaphase analysis and determined that CMA should be the first tier test for developmental defects, such as congenital anomaly, developmental disability. Because CMA provides a much higher diagnostic yield, about 15% to 20% for genetic testing of individuals with developmental delay, intellectual disability, autism, or multiple congenital abnormality, comparing with karyotype. We used to use karyotype G-banded chromosome analysis to make a genetic diagnosis for those patients. After 2010, chromosomal microarray became the first tier of genetic test. Over these years, more than 10 years, the resolution of chromosome microarray has been increased a lot, from detection of microdeletion duplication of about 1 megabase or more, or 100 kb or bigger, to smaller copy number change. Some of them are about 1 kb, and some of them only involve single gene or single exon. Because of the resolution of CMA increased, it is possible to use CMA to detect small copy number change that is associated with autosomal recessive disorder, and to know the features of those autosomal recessive copy number changes. Here, I wanted to kind of say it again. For genomic disorder, the copy number change only affect 1 copy of chromosome for autosomal chromosomes. So the inheritance for genomic disorder is autosomal dominant or X-linked. It can be fully penetrant, like a Weimin Bi syndrome, or it can confer susceptibility to some disease like autism for 16p11.2 deletion. But only 1 copy of the chromosome is affected. But for copy number change that is associated with autosomal recessive disorder, for a patient to get affected, it has to have 2 allele affected. So the 2 allele of the gene have a mutation. If there is only 1 copy of chromosome is affected, mutated, then that individual is only a carrier. He may not have any phenotype, asymptomatic. So autosomal recessive disorder is not a genomic disorder. However, copy number change, those genomic variants, they can cause autosomal recessive disorder. There are different mechanisms for copy number variants to cause disease. For autosomal recessive disease, usually the mechanism is loss of function. CNV can cause loss of function of a gene through deletion of the entire gene or deletion of part of the gene. Like here in the panel C, gene interruption. Those are the major ways for SNV to cause loss of function. Also, there are different loss of function mutations that may cause recessive disorders. We know very well single nucleotide mutation, SNV point mutation, they can cause loss of function associated with recessive disorder. Copy number change also can cause loss of function and lead to recessive disorder. Those copy number change can be small, intragenic, or can be larger, involving the entire gene. Some of them, even large, can involve multiple disease genes. For autosomal recessive disorder, it's necessary to have both alleles mutated. So because either CNV or SNV can cause loss of function allele, there are different ways to combine these two alleles. One allele is SNV and the other allele is CNV. The SNV can be inside the CNV or can be outside the CNV. For the SNV inside the CNV, if you use Sanger sequencing or other method like a sequencing method, detect this single nucleotide change, it is apparently homozygous SNV. But when you do the parental study, you realize only one parent carry the heterozygous deletion. In this situation, it's necessary to do further study. I will have an example to show you this. Another way is CNV combined with CNV to cause autosomal recessive disease. The CNV can be a homozygous deletion, or there are different ways of compound heterozygous CNV. These two CNVs can be overlapping or can be separated. If they are separated, when you do the CMA, you will see this pattern: heterozygous deletion, normal, copy neutral segment, and then followed by heterozygous deletion. In this situation, you also need to do additional study to see the segregation of these two heterozygous deletion in order to make diagnosis. Here is one example to show you that SNV is located inside the CNV. So this patient has severe combined immune deficiency. Sanger sequencing and West whole exome sequencing identified apparently homozygous mutation in the gene IL7R in the patient. Parental study showed the mom is wild type, didn't carry this mutation. Dad has the heterozygous mutation in this gene. Additional study was performed by CMA. CMA showed that the mother who doesn't carry this point mutation actually carry a heterozygous deletion in this region. The deletion include the location of the point mutation. And the patient have two alleles, both mutated. One carry a point mutation, another carry a gross deletion in this gene. This is one example to show you that SNV combined with CNV to make a diagnosis of autosomal recessive disorder as CID. Here is another example. We made a diagnosis. The patient is an infant with features suggestive of Cohen syndrome. This is a multiple system congenital syndrome. What we detected by CMA is two separate deletions. One deletion is 130 kb, involving exons 15 to 19 of the gene VPS13B. Another deletion is also involved this gene, but in a different location, involving exon 24 to 30. The probe between these two deletions showed copy neutral. There is no deletion. So the pattern detected by CMA for this region is deletion, normal copy, deletion. We performed a parental study to see the inheritance of these two deletions. We found the father carried the first deletion, the mother carried the second deletion, and the child inherited both deletions from the parents. These two deletions are in trans in the patient. This is also confirmed by fish analysis. You can see here one deletion region was labeled using a probe, red probe. Only one chromosome 8 has this red probe. Another region was detected using a green labeled probe, and another chromosome has a green labeled probe, but we didn't see any chromosome 8 has both red and green probe. That means each chromosome 8 has a deletion, and the deletions are different. Those two deletions, they are in trans. This example highlights the importance to determine the phase of deletion, normal deletion pattern identified by microarray analysis. If you don't do additional analysis, you think these two deletions are in cis, then this patient would be a carrier, not affected. Copy number change not only cause monogenic autosomal recessive disease, but also involve recessive contiguous gene syndrome. And it may combine with autosomal dominant disorder to form very complex phenotypes. If a deletion affects two or more recessive disease genes, and there is no disruption from an autosomal dominant disease gene, this homozygous deletion will lead to a complex recessive phenotype. We called this disease is recessive contiguous gene syndrome. This is analogous to dominant contiguous gene syndrome. When we see contiguous gene syndrome, we often think microdeletion duplication syndrome. Those only affect one copies of chromosome. For example, Miller-Dieker syndrome involve multiple disease genes. If only this one gene is affected, that will cause isolated lissencephaly. But if the adjacent gene YWHAE is also deleted in chromosome 17, the patient will also have other phenotype like craniofacial features in addition to lissencephaly. Here is one example of recessive contiguous gene syndrome. We actually have an Weimin number for this disorder. This is called hypertonia cystinuria syndrome. The feature is cystinuria growth hormone deficiency and muscle weakness. This disease is caused by homozygous deletion of two genes. They are next to each other. One is SLC3A1, another is PREPL gene. Both genes can cause autosomal recessive disorder. When a deletion deleted both genes, it will cause this complex phenotype, recessive contiguous gene syndrome. The right side, I show you one example how we detected homozygous deletion that causing this recessive contiguous gene syndrome. This is by CMA for heterozygous deletion. The log ratio is -1. But here for this patient, the log ratio is much lower than -1. It is about -5. So we know there is a homozygous deletion in this region. In a previous study by Dr. Lapsky's lab, they checked the recessive genes in the genome and identified 294 genomic regions that contain consecutive recessive disease genes that have potential to be associated with recessive contiguous gene syndrome. The study was performed in 2012. At that time, there are about 1,200 autosomal recessive disease genes identified. Right now, this year, there are about 2,000 autosomal recessive disease genes identified. So it is expected there are more than 300 such regions in our genome. For the detection of autosomal recessive copy number variants, microarray is a way, but it's necessary to keep in mind that for the clinical array, usually it is used for detection of a large copy number change, more than 100 kb. So using this kind of clinical array, you can detect the large autosomal recessive copy number variants. For example, the NPHP1 deletion in chromosome 2q associated with kidney disease. But for smaller one, exonic one, you can't. Those days, there are more and more publications using NGS, next generation sequencing, to detect the small copy number change. And those may cause autosomal recessive copy number variants. And there are also some other ways, molecular ways, to detect the small copy number change, but those, they are not a high throughput, not genome-wide testing. In BG genetics, we also BCM, we have been using microarray, customized microarray to detect the copy number change for about 16 years. The array has been evolving over these years to increase the number of probes and also increase the coverage of probes for the gene associated with disease. One big change for our array design is in version 8, we added exonic coverage for 1,700 genes, including 378 established recessive disease genes. Because of increasing number of probe and also increasing coverage for disease gene, the detection rate is also increasing. One study also performed by Dr. Lapsky's lab showed that with this increasing exonic coverage for autosomal recessive disease gene, it is possible to detect single exon small copy number change in autosomal recessive genes. This study checked the carrier status for copy number change in 21,000 individuals tested by CMA using version 7 or version 8 microarray. It detected 3,000 heterozygous carrier deletions. From this figure, you can see for version 8, the bottom part, the CNV detected are smaller, most of them are smaller than comparing with the CNV detected by version 7 array. Also, most of the CNV detected by version 8 involve single gene, recessive gene, or single recessive disease gene. So, exonic coverage of autosomal recessive gene in an array enable the detection of small single exon copy number change. Now at those days, our array is version 11. That's a comprehensive array, including probes, not only for detection of copy number change, but also detection of absence of heterozygosity. There's a screening for heterozygous absence of AOH. This array has much more gene covered at the exon level. When I say covered at the exon level, it means it has much more probes placed over exon. In average, every exon is covered by about 4.2 probes. And for those intragenic region, intronic region, we put much less probes. And those targeted genes with enhanced probe coverage, including both autosomal dominant and autosomal recessive, we try to include all the disease genes covered with enhanced coverage. From 2006, we started to use adjuvant oligo array to 2018. The time we performed this study, we had about 70,000 patients studied by chromosomal microarray analysis using our oligo arrays. And we have been using this array to detect the copy number change range from exonic copy number change to MAC base level. Some of the autosomal recessive copy number change that contribute to diagnosis have been identified, and we publish those cases as a case report. However, we haven't at that time, we haven't had a systematic study about the disease causing autosomal recessive copy number change. That's why we performed this retrospective study to see the contribution of copy number change on autosomal recessive disorder diagnosis. In addition to the patient referred for chromosomal microarray analysis, we also include in this study the patient we performed exon study, because we performed the copy number change for those patients too. One is use the NGS data, another is use the C-SNP array. C-SNP array, complementary SNP array, is the array we use for every single patient we perform the exon. So when we received one patient sample, we split the DNA sample into two aliquot. One aliquot is used for exon NGS data, and we use the NGS data to analyze detect the homozygous, hemizygous deletion. For another aliquot, we use it for C-SNP array analysis. That's an Illumina array with about 240,000 probe. At this time, we use another version is 550,000 probes. This SNP array, the major purpose is to serve as a quality measurement to ensure correct sample identity and to assess the NGS sequencing quality by comparing the result from the SNP array with the NGS data. However, this array can also be used to detect copy number change, large copy number change, heterozygote, homozygote, or hemizygous. The advantage for this SNP array is we can detect a large copy number change. We did a comparison study which found that this SNP array can detect about 70% of disease causing copy number change identified by comprehensive microarray. Those small exonic copy number change that can be detected by comprehensive microarray cannot be detected by this complementary SNP array. And this C-SNP array is also wonderful for AOH detection, very high resolution, high reliable. The limitation for this C-SNP array is this Illumina SNP array is not designed for clinical copy number change detection. So smaller one, sometimes even 2 meg base duplication, we cannot reliably detect by this SNP array. Here is one example. We use this C-SNP array to detect copy number change and make a diagnosis. This patient has infantile spasm. Using the C-SNP array, we identified one homozygous deletion, about 2.2 kb in the patient. This is a trial case. For trial case, we perform SNP array not only in the proband, but also both parents. The SNP array called heterozygous deletion in this region, WWOX gene. The mother has 113 kb deletion heterozygous. The father has 87 kb deletion heterozygous. these two deletions are not the same, but they overlap. The overlapping region is the exon 6 of WWOX. That's the region where we identified homozygous deletion in the proband. So this patient has compound heterozygous deletion in WWOX, which lead to the autosomal recessive disorder, epileptic encephalopathy. This is an example that two deletions, they are overlapping. We actually published one paper about using cis-SNP array to increase diagnosis in ES. We studied about 11,000 ES exome cases. Using the cis-SNP array, we detected 357 findings. Those are diagnostic findings. Most of them, if not all, most of them are heterozygous deletion, single copy, copy number change. Some of them are UPD aneuploidy. Some of those West cases also have CMA. We checked the copy number change, disease-causing copy number change, detected either by cis-SNP array or CMA. We identified 3.3% individuals have this pathogenic finding. So with copy number change detection in exome, it can increase the clinical diagnosis. Okay, here in this study published in the paper, we included two cohorts. One is CMA, about 70,000 patients. Another is exome, about 12,000. We retrospectively identify those autosomal recessive disease causing copy number changes in this patient. I need to emphasize that we also have lots of autosomal recessive copy number change identified in patient. There's only one copy of mutation identified. For those, we didn't include them. We only include in this study the autosomal recessive copy number change that we know definitely contribute to the diagnosis with both mutations in both alleles identified. We identified 87 cases with such disease-causing autosomal recessive copy number variants. 17 cases have mutations of CNV in both alleles, and 17 cases have a combination of CNV with SNV. For those 70 cases with only CNV contribution, we found that homozygous CNV in 63 cases, about 90%, and 7 cases with compound heterozygous CNV. Here is the pattern of mutations identified in these 87 patients. So as I mentioned, 63 patients have homozygous CNV. 7 patients have compound heterozygous CNV, and 17 has CNV combined with SNV. Most of those patients have deletions. Only one homozygous CNV is duplication, and one compound heterozygous CNV is duplication. we checked the size distribution, the number of genes, and also exon content of those AR CNVs. Here is the size distribution. You can see majority of autosomal recessive CNV, they are bigger, they are smaller, they are smaller than 100 kb. The smallest one is only 68 base pair single exon deletion. None of homozygous deletions are bigger than 1 megabase. We only identified 4 cases with heterozygous deletion larger than 1 megabase. You know, the bigger the size of the CNV, the more likely it involves more disease genes. That's why homozygous may cause too severe phenotype, and the carrier of such homozygous deletion cannot survive. So majority of autosomal recessive CNV, they are small. They can't be detected by microarray if there is no enhanced coverage for these autosomal recessive genes. Here is the gene content in autosomal recessive CNV. In total, there are 157 such CNVs. We can see 94% of those CNVs affecting only one single gene. And about 97% of homozygous AR CNV affect one single gene. Only a few cases we detected that affect multiple genes. Exon content of autosomal recessive CNV also showed that most of autosomal recessive CNV, they only involve part of the gene. About 41% of AR CNV affect a single exon, and 35% affect multiple exons. The conclusion is also the autosomal recessive CNV may not be detected using a microarray without enhanced coverage in autosomal recessive disease gene. Since in this study, we included different type of CNV analysis, including microarray, C-SNP array, and NGS data, we performed a comparison to see the detection sensitivity for those different methods. In the right side, you can see for both heterozygous CNV and for homozygous autosomal recessive CNV, CMA with enhanced coverage for autosomal recessive disease gene showed excellent sensitivity, about 90% or higher. But for C-SNP array, which doesn't have good coverage for autosomal recessive disease gene, showed the poor sensitivity. NGS data from West actually is very good for detection of homozygous deletion. The next part is we checked which genes are affected by AR CNV. Genes are variably affected. Here you can see majority of the genes only affected in one patient, 49 of those genes. For the other 8 genes, CNV were detected in multiple patients. TANGO2 gene is the most frequently affected gene. We identified homozygous deletion of TANGO2 in 6 patients, identified compound heterozygous CNV in 1 patient, and also detected 1 compound heterozygous CNV plus SNV in TANGO2 in 1 patient. For 87 patients that have the autosomal recessive CNV, 63 have homozygous AR-CNV. So we wanted to know what result homozygosity in this patient. We have the homozygosity mapping data in 57 cases of homozygote CNV. We identified large LH encompassing the CNV in 40 cases, about 69% of cases. Total LH in these cases, for each case, ranged from a single LH of about 7 MAC base to multiple chromosome, multiple region, about 400 MAC base. And some of those patients apparently have the homozygosity because of consanguinity. They have over 50 MAC base total LH. This is identified in about 50% patient. They have consanguinity of second cousin level or higher. Interestingly, we have 17 cases that didn't have large LH encompassing the CNV. I took a closer look at these 17 cases. 8 cases actually have CNV that have high frequency, high allele frequency in the population. And 4 of them have HBA1, HBA2 gene deletion. We know this deletion is frequently seen in Southeast Asia. The carrier frequency is around 1 out of 25. Another 2 is NPHP1 gene deletion, STRC gene deletion, which is associated with hearing loss. The carrier frequency is also relatively high. So homozygosity in those patients is because of a high carrier frequency. And we identified 4 patients with TANGO2 exon 3 to 9 deletion, and that at that time we didn't detect apparent LH over this TANGO2 gene region. However, when I prepared this presentation, I took a closer look at the raw data. There are actually 0.8 to 2.1 MAC base LH encompassing the TANGO2 gene region.
Weimin, sorry to interrupt, Weimin. We have 4 more minutes.
Okay, thank you for the reminding. So this TANGO2 deletion is due to distant shared ancestry. For these 4 patients, 2 of them are European Caucasian. A previous study on 3 families with this deletion showed that this deletion is likely a single ancestral variant in the European population. The study identified the shared 20 variants in the 350 nucleotide region around the breakpoint of this deletion. The frequency of this deletion is much higher in the European population comparing with the other population. For the other CNVs in the homozygous, with the homozygous deletion, but without the LH, we a closer look also identified the LH, except for the TBCK exon 23 deletion. This deletion is a recurrent deletion that was seen in multiple different populations. So in summary, the source of homozygosity in homozygous AR-CNV is because of consanguinity, high carrier frequency, or distant shared ancestry. Autosomal recessive CNV also contribute to multiple diagnoses. A study showed that about 5% of cases positive by West has multiple diagnoses. In this study, we found those cases with diagnosis from CNV, they have 2 or more molecular diagnoses in 19.5%. So the percentage of the patient with 2 or more molecular diagnoses is much higher in this cohort. Autosomal recessive CNV also directly needed to 2 diagnoses in 7 cases, including 2 cases that affect 2 autosomal recessive disease genes adjacent to each other, causing contiguous gene syndrome. We also identified 5 patients with large copy number change, causing microdeletion syndrome, and unmasked the recessive mutation in the other allele. One of those patients, I'm going to give you detailed information. This patient is a newborn with prenatally detected pathology of failed heart disease. We performed CMA on this patient using comprehensive array. We identified the DiGeorge syndrome deletion, about 3 meg base typical deletion. We also identified a homozygous deletion in TANGO2. The TANGO2 gene is located close to TBOX1 gene inside the DiGeorge syndrome typical deletion region. TANGO2 gene is associated with an autosomal recessive disorder. The name is very long, recurrent metabolic encephalomyopathy crisis associated with rapidolysis, cardiac arrhythmia, and neurodegeneration. One important information is those patients, they are prone to metabolic crisis in time of fasting or stress. So medical management is very important, that the patient should avoid triggers for accurate metabolic crisis. We also performed a parental study on this case. The maternal CMA detected the exon 3 to 9 in TANGO2 deletion. So this patient has 2 chromosome 22, 1 chromosome 22 carry the DiGeorge syndrome deletion, another carry the TANGO2 deletion inherited from the mother. This is a compound heterozygous deletion, AR deletion. And this is also a good example to show you that a precise and complete molecular diagnosis can be reached through microarray detection of a large deletion plus an exonic deletion in an autosomal recessive disease gene. Summary. Our study showed that both CNV and SNV analysis are necessary in order to provide a precise genetic diagnosis, because CNV contribute to the autosomal recessive disorder, either with or without SNV. CNV may cause recessive contiguous gene syndrome or lead to complex phenotype with dominant disorders. Multiple diagnoses are more frequent in cases with autosomal recessive CNV. Without the small CNV detection, you may miss some of those diagnoses. A microarray is necessary to have enhanced coverage in autosomal recessive disease gene to detect the small AR-CNV, because most of the AR-CNV, they are smaller than 100 kb involving single gene. Those most AR-CNVs, they are homozygous deletion. So if this gene is well covered in the array, those homozygous deletion can be easily detected. I will stop here. Thank you.
All right. We have one question from Joseph. I think that's a good question also. Joseph, can you unmute yourself and ask the question? I think you are muted. Joseph?
It's okay.
I'm wondering how the enhanced bio-nano. Oh, am I muted?
Your audio is not great, Joseph. We'll ask for you. So he's wondering how this enhanced CMA would compare to a long read sequencing method, what like bio-nano?
I have limited experience with bio-nano. My understanding is bio-nano can detect the sequence, right? Single nucleotide sequence, but probably the mutation rate, like the false detection rate, is high. For me, currently we don't have the experience on using nano technology, this technology to detect the CNV.
Okay, great. Well, thanks for the excellent talk. Great overview and awesome data from a large data set. So I think everyone, Weimin said that you could email her if you had further questions, right?
Yeah, I think there are many other ways to detect the CNV. However, probably right now they haven't been applied for the clinical diagnosis.
Yeah. Yep, exactly. Okay, well, thank you very much.
Thank you. Bye.